And this is a very important section for us as individuals because this is about regulating the cell cycle, making sure the cell cycle doesn't go on if it's dangerous for the organism or if the cell is not completely ready to do so. And one of the big things that's going to regulate a cell cycle, one of the ones we know the most about, are these cyclin and cyclin-dependent kinase proteins. So your objectives for this section are to be able to summarize the role of cyclin proteins in controlling the cell cycle, to explain how cancer relates to the cell cycle, talk about apoptosis, and we're going to talk about stem cells too, because they are involved in cell cycles, and they are important, and they have potential to be very important in our lives. So we're talking about DNA still, and the DNA is made up of nucleotides, A, C, T's, and G's, and those four letters are very important, and the order of those four letters in DNA is very important. And when we don't get the letters right, or when the cell doesn't get the letters right, we call it a mutation. There are things we do know that regulate the cell cycle would include cyclin and cyclin-dependent kinase. Again, if an organism has cells that are not being controlled well in the cell cycle, we can end up with cancers. And we know some things that lead to cancers, make us more susceptible to cancer, and we call those carcinogens. And then some of the other aspects we'll talk about are apoptosis, which is a genetically programmed cell death, a very different kind of death than injury death, also called cell suicide. And then we'll talk about stem cells, which have the potential to treat a lot of human diseases, and they are important in our everyday life. They replace our skin constantly, they replace our blood constantly, and we all started out life as a single stem cell. So we talked about coming to an intersection as a driver, and we can think about the cell cycle as having these intersections as well. There will be places along the pathway going through interphase, mitosis, cytokinesis, where we would have a stop sign, so to say. And at that stop sign, the cell will look around, make sure everything's ready, and proceed through the stop sign if it's safe, or do something else if it's not safe. And one of the things we know that can control that are the cyclin and cyclin-dependent kinases. Two different proteins, but they're both very much involved in controlling whether or not that cell moves through these stop signs. So we need two signals. One of them is a cyclin protein. The other is a cyclin-dependent kinase. So cyclin is a protein. The protein, which has instructions in the DNA, it's written in a specific order of nucleotides in the DNA. And this protein is only made if the cell is ready to move through one of the stop signs. So cyclin, if we go back to our driving analogy, would be like your foot on the accelerator. Your foot is not always in the vehicle. The accelerator is always in the vehicle, and you only put your foot on the accelerator if you're ready to move forward. So in this case, when a cell is ready to move forward, it doesn't put its foot on the accelerator. It goes into the nucleus, turns on the genes for cyclin. The cyclin messenger RNA is copied. That messenger RNA will be turned into a cyclin protein, and that cyclin protein will interact with cyclin-dependent kinases. These cyclin-dependent kinases are like the accelerator in your car. They are always there, but they're not always active. This case, it's not a pedal, it's an enzyme. It's a chemical that speeds up other chemical reactions. And the reaction that all kinases carry out is to add a phosphate to another protein. And adding a phosphate to other enzymes will have one of two effects. It will either turn them on or turn them off. And you can imagine that to move for a cell, the complex nature of a cell, to move through a stop sign, there are things you need to turn on and things you need to turn off. And that's exactly what cyclin-dependent kinases do as long as cyclin is present. So where are these checkpoints? Where are the stop signs? These combinations of cyclin and cyclin-dependent kinases come into play at the start of nuclear division. And hopefully you all remember the start of nuclear division is mitosis. So before prophase starts, you're going to have to produce some cyclin. That cyclin will bind to cyclin-dependent kinases, and cyclin-dependent kinases will turn on and off the machinery that's needed to get rid of the nuclear membrane, change the chromatin to chromosomes, move the centrioles where they need to be, set up the spindle fibers. At the end of G1, there's one of these checkpoints. Some cells can't get through this checkpoint. If you're a heart cell, a nerve cell, or some plant cells, you don't go past G1. But if you can, at the end of G1, when you are told it's time to divide, you produce cyclins. Those cyclins bind to cyclin-dependent kinases, turn on and off the machinery that's needed to go into S. And that's another step, another place where there's a stop sign, the DNA replication process. This is very tightly regulated. There's only one time in the life of a cell when all the DNA is made into a DNA copy, and that's the S phase of interphase. And at the end of that, there's a stop sign to make sure that it's all been done correctly, because you absolutely need to have two sets of instructions if you are going to build two new cells. There are other cyclins and cyclin-dependent kinases that drive protein synthesis for G2 and the end of G1. They're all places where it's very important to make sure that the cell is ready, able, and safe to move forward in the cell cycle, just like when you pull up to a stop sign when you are driving. Because if we don't control this well, we can end up with uncontrolled cell growth. And uncontrolled cell growth is what we call cancers. Cancers crowd out normal tissues. There's no extra space in your heart, your brain, your liver, your kidneys for even small amounts of extra tissues that aren't doing what they're supposed to do. The structure of your heart, your brain, your kidneys, your liver are all very important, and if you put other stuff in there, it messes up that structure. Another big difference between cancer cells and normal cells is cancer cells use up nutrients. They will take nutrients away from healthy cells. You can eat a well-balanced diet, but your cancer cells will get the nutrients before normal cells. Now, if we use the analogy of a car or stop sign for controlling the cell cycle, you can imagine that your car has a lot of ways you could accelerate. It has a lot of ways you could stop it from going anywhere. And you always go get your car repaired when there are problems. And the same kind of thing is true with cells that become cancerous. One single change is not going to make a cancer. It's going to take a lot of mutations in a lot of different genes before you get to the point that cells grow uncontrollably. That takes a long period of time. Most cancers don't show up until people are in their 40s, 50s, 60s even. Some of the factors we know. Some of the factors also include genetics, and some genetics you can inherit. There are a number of cancers that run in families because they have mutations in some of these genes already. But you need more than one mutation. It takes a long time to accumulate enough mutations to get cancer. But we also know, aside from genetics that we're born with, mutations can happen in the DNA because you interact with things in the environment. And those things in the environment that can cause mutations in the DNA, which might cause mutations in genes that control the cell cycle, are called carcinogens. Cigarette smoke contains a huge amount of carcinogens. And there are other things that we just can't avoid, like sunlight, that is carcinogenic. All right, so normally when a cell divides, there are a couple things it can do. We talked about these in class as well. And one is if you take one cell, divide it into two cells, one of those two cells might die. That's called apoptosis. It's a programmed cell death. That means you can, even though you get two cells from one cell every time you divide, only one of those cells survives. There are, that is one way to control the total number of cells in the body, but it's also used during development so that we get the shape of our bodies, the shape of other organisms' bodies. Apoptosis is an important part of developmental biology, and it's a possible way to help control excessive cell numbers to prevent cancers. The other type of cell that we'll talk about is called a stem cell. A stem cell you can think of as a potential cell. It has the ability to become another type of cell. So in this case, when a stem cell goes through mitosis and you end up with two cells, one of those two cells will stay a stem cell. The other one will grow up to be something else. We have stem cells in our skin. So if we need more skin cells, we divide a stem cell. One stem cell stays. We never run out of stem cells. The other cell becomes a skin cell, makes its way to the surface of our skin. We have blood stem cells that can make all of our red blood cells and white blood cells. In our intestines, stem cells are very important cells. These are all examples of adult stem cells. They can become one or two, or maybe even a few different kinds of cells. And they are useful if someone needs a bone marrow transplant. We only have to give them bone marrow stem cells, and we can replace all of their blood. That's one example of how we use stem cells to treat diseases. The other type of stem cell is an embryonic stem cell, and an embryonic stem cell has the ability to become any other cell in the body. We all started out life as one embryonic stem cell, and that embryonic stem cell gave rise to all of our cells: brain cells, heart cells, liver cells, skin cells, blood stem cells. And if we could use those embryonic stem cells to treat diseases where cells can't multiply, like brain cells, heart cells, they could become very important. Unfortunately, the source of embryonic stem cells, the easiest source of embryonic stem cells currently, is from an embryo. And an embryo is a potential human being. So there's a lot of controversy surrounding the use of embryonic stem cells to treat disease. So in summary, the cell cycle of eukaryotic cells is regulated by cyclins. It's regulated by cyclins because there are checkpoints along the way that are places where the cell stops, takes a look around, and makes sure it's ready to move forward. If it's ready to move forward, the cyclin-dependent kinases are already there. The cell produces the cyclins to get the cyclin-dependent kinases turned on. We have to regulate the cell cycle very closely because we know when it is not regulated, one of the things that can happen is cancer. And cancer is simply defined as uncontrolled cell growth and division. Apoptosis is a programmed cell death, very important in developmental biology, plays a role in controlling cancer. And stem cells are unspecialized cells that become specialized cells, like skin cells, with the proper signals. And they come in two forms: embryonic, that can become anything, and adult stem cells that can become a limited number of things.